big topic. So I'd like to thank Kavita and Christian and Sam, who's not here, for inviting me to this program. So there's a quite humbling list of co-speakers, but I try to present you something that makes sense, hopefully. So I think I'm going to sort of catch up where Mike left it and move on to models that are more specifically about adaptation and trying to see how we could study that at the levels of traits or at the level of fitness. I try to make this a bit more explicit. So just start very simply by what we mean by adaptation, because even within evolution, people mean different things by adaptation, at least in biology and within evolution. Some people refer to a state like adaptation is being adapted, okay? So there's a match between some phenotype and some environment. Lots of people in macroevolution, they're talking about traits. Lungs are adaptations to terrestrial life. So it's a numerable thing, it's an adaptation. So it's a discrete thing, you know, but usually here people in this room, when they're talking about adaptation, what they mean is the process. So they talk about adaptive evolution. So we could define this as an increase in fitness through a natural selection that's causing a genetic evolution of traits that sort of match the requirements of this environment. So ideally, we want to have all this in mind: the environment, the traits, genotypes, fitness, right? That's, to me, the basic ingredients to study adaptation. Actually, lots of people talk about adaptation at the individual level. So physiological adjustment, phenotypic plasticity, all these things that we're not going to talk about here, although they're super important when you place an organism in a new environment, and that also affect populations. So I'll try to talk about that in my final talk at the end of the program, but that's not what we describe as adaptation. So we use a different word for that. So of course, some examples, lots of people here know about this, but perhaps some don't. So you have this kind of textbook example of this Darwin's finches in this island in the Galapagos that Peter and Rosemary Grant have been studying for 40, 50 years. And so there are different species. There's a big whole story about this, but this medium ground finch, for instance, it seeds, and they have shown that different phenotypes in the population, some are larger and have larger beaks, some have smaller beaks, and they specialize on different sizes of seeds, in the and these also change with the environment, the precipitation, etc., the proportion of the different types of seeds. They have been able to connect these different selective pressures to how the trait changes over time, and also how selection on the trait changes over time. So here is one of the cases where this is an island. You can stay there and observe everything. They sort of have this whole story, even the genes involved in that. So this is why it's now a kind of textbook example. Another example that, unfortunately, I think people are very familiar with here. I was reading that nearly 50% of Staph aureus are multi-resistant here in India, actually. So it's a big issue. It's a big issue worldwide, but antibiotic resistance has become kind of also a textbook example of adaptation because you can, one of the reasons is that you can study it in the lab. So in this experiment, they made this, this kind of big Petri dish where you have the gradient of antibiotic concentration. So it goes up and then down here. So you have these two gradients. And then they inoculated the population here. So at first, population grows here of these E. coli, and then they're not able to grow in the next environments because they're not adapted to this concentration of antibiotics. And then some mutant is able to spread here after 88 hours. And then little by little, they are able to colonize this higher and higher concentration of antibiotics through stepwise mutations that confer them resistance to these things. What's interesting in this story is that here have this dynamic aspect, then they track the dynamics through time. And also they were able to track all these different genotypes that were selected. And also to measure their antibiotic resistance independently. So for instance, here, this red one here was already adapted to a higher dose of antibiotics than what it evolved for. But other than that, you see that kind of progressive increase of antibiotic resistance in this region here. They're mostly adapted to this range of doses, etc. So in this case, you could track the dynamics of adaptation, also the genetic level. Well, not because Christian is organizing this, but I sort of have to cite this because just thermal adaptation is a big, big issue. And especially in the context of climate change, lots of people are interested in whether species will be able to track changing environments, whether they're close to their thermal limits or not. So it's been a big field for a while in ecology and evolution. Here, it's an experimental evolution example where in Christian's group, they adapted these Drosophila simulants to these patterns of cycling temperature. Daily cycles are pretty large, reach high temperatures. And they observed an increase in fecundity. So there's been adaptation at the level of fitness. Then they were able to track genetic changes throughout the genomes. And then you have these different trajectories of allele frequency change. And this will come back during the meeting, I guess, because this sort of sparkled some discussions about these polygenic responses. There are redundant loci that respond in different replicates. Okay, so from this, I mean, if you'd like to study adaptation from the basic ingredients, what you need is you find which phenotypes are favored in which environments for all traits, if you can, or the relevant traits. Then you need to decipher the genotype-phenotype map because you want to know how selection of the trait propagates to its underlying genetic basis. And then you need to do what we heard from before. You derive the population genetic change that are caused by natural selection, plus these other forces like drift that we heard a lot about, but also, you know, and mutation, but also recombination, migration perhaps. And then from that, you can go back to infer the evolutionary changes in traits, response to selection, and the changes in fitness, which is what you're actually interested in most cases, in this field at least. Of course, what I want to emphasize here is that I think the rest of the discussions in the next 2 weeks will be mostly about evolution of traits, which is interesting, of course. But there's also lots of work in population genetics that bypasses these steps that involve the phenotype and the environment, because they're more difficult to get than sequencing a lot. So lots of people focus on effects on fitness. And so the goal in the overview of this lecture is how can we understand and predict the dynamics of adaptation, really? This is the general question. And I'll start by approaches that focus only on fitness. And I'll just give some snapshots. So I won't really go into the details of each of that. So it can be seen as kind of an introductory lecture, I don't know. Different regimes. So there's a regime of low mutation where really you have sequential fixation of mutations. Then you have adaptation in more polymorphic populations. And this will lead us to this distribution of fitness effects that we heard a little bit about, but we were just focusing on the deleterious ones, which are not the ones we're interested in here for adaptation. So we'll talk about the whole distribution. And then from there, we'll move on to approaches that account for the phenotype. And the transition will be how the DFE changes when you include some traits under selection. And then actual models of phenotypic evolution. So this is the kind of argument I want to lay out. I hope I have time to go through all that today. The second lecture will be about having an explicit environment and then models of adaptation to the changing environment. Okay. So adaptation at the level of fitness. Well, fitness, of course, it's kind of hard to define fitness in a course on about evolution, but using the word, so I'm going to just define it as kind of the engine of selection. Is like this very central evolutionary concept. It's a measure of the contribution to the next generation in the broad sense. Most evolutionists, we focus on relative fitness. We're only interested in competition between genotypes and phenotypes. That's what matters. But when you want to estimate that, you often need some measure of absolute fitness, population growth. And it's these differences in absolute fitness that lead to relative fitness in the first place. So this definition is kind of simple. When you have non-overlapping generation, kind of right Fisher population, it's just survival times fecundity. But even there are some debates about, well, over a complete life cycle, it should be from zygote to zygote. Mike raised this point. You don't often measure things at the level of zygotes. But it all becomes more complicated when you have overlapping generations, which is the case for lots of the animals and plants or continuous time. In this case, there's a benefit to reproducing earlier. So it's not just how many offspring you leave. This has been studied a lot by quite a few people, but this is the first time I'm bringing this Fisher. It's not the Fisher who was sitting here just before. It's another one, slightly known to in population genetics. Is a statistician you might have heard about who did PopGen, invented PopGen as a hobby in his spare time. And so in his book, he had some equations. He also bothered to define fitness based on life tables in humans, etc. And so he used this, what's called the Euler-Lotka equation to define fitness. But here I'm representing this concept of reproductive value that he introduced. I think it's it's the contribution of individuals of a given age to the future population. So, and this is just to emphasize that, so contribution will depend on the probability to survive to later ages weighted by their fertility at this age. And then it's discounted by this effect of the fact that the population is growing. So, and this is kind of the usual population genetic jokes that everything seemed obvious to Fisher, and then people had to demonstrate it. By the analogy of compound interest in the present value of offspring is easily seen to be in this. So this is the kind of the idea of discounting for what happens later. It's kind of gets less interest from that. So that's what was obvious to him, I guess. So then he could show this graph of how reproductive value changes with age. This is just to illustrate that fitness can be more complicated. There are other conceptual issues, practical issues with fitness. Theoretically, it should be a propensity. It's not, you cannot really define the fitness of an individual in theory. It's a kind of, fitness should be the expectation of a random variable, and an individual is just a realization of that. So it actually includes some elements of demographic stochasticity that contributes to drift, actually. So not everyone accounts for that, but then, of course, all these ecological aspects, probably we won't talk about this, but even the relative fitness, they may change with population size. And this is like density-dependent selection. For instance, you could have rapid colonizers that are favored at low density, but good competitors at high density. And also relative fitness may depend, it may not be a property of the phenotype or the genotype, it may be a property of interactions among genotypes. This is frequency dependent. So here we're gonna sort of neglect all that, but I still wanted to mention it because even people studying adaptation, they're interested in that a lot. And it shows up in experiments, like in Lenski's experiments. All these things, even when you design an experiment like a model, it doesn't want to do what you tell it to do. So anyway, we'll focus on density and frequency independent adaptation. Also, well, you might wonder, why do you even want to study evolution of fitness, you know? Why? Well, it's kind of the evolvability of fitness is kind of setting the upper bound for evolvability of other traits because they're selected through their covariance with fitness. So that's one reason. Another reason is sometimes the rate of change in fitness is what matters, especially in cases where you link evolution with demography, and when the rate of change in fitness will affect whether a population is able to grow or not. And so this is something that, Mike Lynch has worked in one of his numerous previous lives. And others have worked on that. So, well, in this scenario where there's an abrupt change in the population would go extinct without evolution, but if it evolves fast enough, then it restores a positive growth rate, it avoids evolution. If not, then it might reach this region where bad things happen to them because of even other reasons like demographic stochasticity and things like that. So this was a theory in the 90s that's been tested empirically in the lab a lot in the 2010s. And so this is one experiment. I just showed it because it looks a lot like that. So they started either from clonal populations or for more mixed, like mixture of clones. They saw that they obtained more rescued under like antibiotic resistance when they had more genetic diversity. So this kind of simple prediction from that. Okay, so now moving on to the actual topic, now that I sort of defined what we're working on. So when you're focusing on adaptation only at the level of fitness, there are different regimes you might be interested in. And so in the low mutation regime, this has been studied a lot. So this has been described as either a strong selection weak mutation framework of Gillespie. There's been a nice review a few years ago that called that origination fixation regimes. It's basically a region where each mutation is, when it fixes, it's in a non-polymorphic population. So adaptation might involve multiple genes eventually, but it's not polygenic because it's only the sequential mutations that fix. And so in this case, adaptation can just be treated as a Markov chains between different fixed states. That's what they illustrated here. You have a mutation that arises, it manages to fix, and then another one arises but gets lost by drift, just by chance, and then this other one. And so there's a time scale where this happens essentially instantaneously as compared to the time between different events. And so you can just focus on the events of fixation and treat this as these fixation probabilities as transition rates. And so there's been a productive field of using that. I think most molecular evolution theory, the way I understand it, is really based on this untold assumption. So if you assume that only beneficial mutation can fix, then you can get a rate of adaptation, in haploids I wrote, yeah, in the simplest case. So the rate of change in fitness is just the input of mutation, which depends on population size times mutation rate, genomic mutation rate, times this integral here, which is like, you have the selection coefficient of mutations, so defined just as in the previous talk. The wild type has fitness 1 and the mutant has fitness 1 plus S. And so, multiplied by its fixation probability, integrated over this thing here. And this is like the distribution of the fitness effects. There could be a range of beneficial effects. So you have distribution of fitness effects for these beneficial mutations. For small positive selection coefficients, it's a well-known result that the fixation probability can be approximated as 2S. So this goes back to one of the founders of population genetics who finished his career in this country, I think, Haldane. And so if you replace that here, you end up with this mutational input times something that depends on the expected square selection coefficient. So here you have something that tells you that there's something about the distribution of selection coefficient of beneficial mutation. Their squared expected, their second moment basically is important for adaptation. In the high mutation limit now, by this I just mean adaptation from standing variation. You don't need to wait for a mutation to introduce new adaptive variants. There's standing variation in the population when selection starts. Then now fitness change over time becomes changes in frequencies of existing alleles. This is like the basic kind of population genetics, original population genetics, let's say. And so now if you consider multiple biallelic loci, and which I still present haploid selection just because it's simpler to illustrate. And it acts independently, so there's no LD, there's no association between loci. And then the beneficial allele at locus I has a frequency pi, and the other allele is qi. Then the frequency after selection is just, the frequency weighted by its fitness divided by mean frequency. And from this, you get that the change of the selection you can approximate as SPQ. So lots of people are maybe familiar with that. It's very easy to show. So now, going back to what we're interested in, the change in mean fitness per generation, then, you can just write as the changes in frequency times the influence of change in frequency on fitness, partial derivatives, which is just the selection coefficient in this model. And so you end up with this formula. It's just the sum of S squared times PQ. So if you treat fitness as locus I as a random variable, which takes these different values depend on the state, which are the alleles, then actually this here is just the variance at this locus, right? The variance, the contribution of genetic variance at this locus to variance like this. If you sum across loci, you recover that change in fitness is just the sum of this VI, which is this additive variance in fitness. And probably it will be explained much better in later courses. But here we have Fisher again. So also known for being quite humble, he called this the fundamental theorem of natural selection in the same chapter as where he defined the reproductive value. The rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. So this is this. Then people have written, there's been some exegesis, how do you say that? There's been some people try to understand what he really meant. Fitness cannot increase forever, right? Because it's compensated by environmental deterioration, which he mentioned, which is any elements of changes in the population or in the environment that's causing fitness to actually remain perhaps unchanged, but still the effect of selection has been to change it, and then something else happens. You can also just write this in terms of allelic effects of these different loci. If you sum this across these loci, and then you can see that this is well, you have NL, the number of loci, times this expectation of this. So where basically expectation of PQ is the mean polymorphism at this loci, the expected heterozygosity, but I'm talking about haploids here, but it's just the diversity, the probability to pick two different sequences when you pick them at random. So it's the measure of genetic polymorphism, the locus, times their mean squared effects of segregating alleles. So here again, we encounter something about the distribution of effects, but it's not exactly the same. It's the distribution of effects on fitness of segregating alleles. So what I want to say with all this is we encounter again and again something about this distribution of fitness effects. So if you're only focusing about fitness, this is what you want to know about if you're trying to predict adaptation. Okay. So the distribution of fitness effects, Mike was just saying that this is measured very crudely, especially perhaps for very small effects. But the approach to do that would be to get some mutants. So either you do spontaneous accumulation of spontaneous mutations under relaxed selection by going through bottlenecks. I think in Mike Lynch's lab, they have been doing that a lot. So I just cited his book here, but you can find some other papers on this one. You can also do random mutagenesis with a mutant. And now there's also some people interested in doing target editing of gene with CRISPR-Cas9, for instance, and just do all the possible sequences and insert them. So this was in that recent paper. Then once you've done that, you either measure absolute fitness. Lots of people working on animals and plants, they would do that. They would measure survival fecundity and try to get a measure of fitness. People working on microbes, they would rather use fitness assays where you compete genotypes against each other. And nowadays, they will mostly sequence them to get their frequencies at the end. And you can do that through bulk competition. You put all genotypes together and then you infer their relative fitnesses. Also, this could be inferred indirectly from patterns of molecular evolution, just like we heard in the previous talk. You use the side frequency spectrum. You try to extract something about distribution of fitness effects. Usually it's about deleterious effects, though. This modification of the so-called McDonald-Kreitman test, but that account for the side frequency spectrum, usually they're focusing on deleterious effects. So not really what we want to focus on today, but still, so this distribution, when you look at them, they mostly include deleterious mutations, of course. So that's kind of the typical pattern. So this is an example in E. coli, mutations in the antibiotics resistant gene. So usually that's how you screen mutants. You put some antibiotics, you just get the resistance, and you look at what mutations there are. This is like a mutation accumulation experiment in phages. And so they usually have this pattern. So mostly deleterious mutations. This is 0, so from here, it's all deleterious. It's usually left skewed in this way. Then you have this excess of, you have this fraction of lethals that have fitness that basically they cannot grow. And then you have some rare beneficials here at the right. Here we don't really see them. So the beneficials, as I said, these are mutations that matter for adaptation. And so how can you study them? Well, one way is to say that they're in the tail, usually, of the distribution, because they're rare and they're on the right. And so you can use extreme values theory to analyze them. I think this was introduced by Gillespie in the early 80s, and it's been used a lot by Orr after that. So the point of this theory, which is not, well, it's not coming from population genetics, it's been designed before, is that many distributions, they will converge to the same family of distribution in their tails. It's called the generalized Pareto distribution.
Whose theorem is that?
Sorry?
Who invented that? Who showed that theorem?
I don't know.
Fisher.
Again? Oh, no.
The Trinity theorem.
Okay. So it could be called the Fisher distribution, but it's generalized Pareto. I didn't know that. Okay. And so there are different domains to which these things might converge, depending on the type of distribution. Lots of distributions that are unbounded, they converge to this Gumbel type, it's called, where its distribution is exponential in the tail. This is what I sort of pictured here, like schematically. Declines exponentially as long as you remain in the tail. And this distribution is expected. What made it powerful, I think, in this Gillespie mutational landscape model, so-called model, is that it doesn't depend on the rank of the wild type or its fitness or anything. It just depends on whether it's in the tail or not, as long as you're in this domain, which we'll come back to later because it doesn't have to be if there's an optimum. Okay, so people have tested that. For instance, in this paper, they used mutants isolated by this so-called fluctuation test, which is a way to detect mutants by imposing antibiotics. And so here their argument was that the distribution they got for beneficial fitness effects, so they get these mutants and then they put them back in the control medium, and so they can have the distribution of fitness effects. They said it wasn't that different from exponential. I let you judge that. I don't know if they had lots of power to check this because there weren't that many. Here they had more. So this is a case where they used this barcode sequence, 20 base pair barcode sequences. So they had hundreds of thousands of them. And then they inserted them in some plasmids, and then they transformed some yeast libraries with that. And then this allowed them to track new mutations that would arise in the population, and they would hitchhike some of these variants. So it's a different way of doing things. Is not generating the variation, is letting the variation arise spontaneously. This is not that different from what Christian and someone did, the Imhoff and Schlötterer paper some time ago, wasn't it? With microsats. So it's kind of the more wider scale method of that. So you just see mutation that arise. And in this way, so they track the frequencies of these barcodes with the assumption that they each carry different mutations that arise by chance. So and then you have all these frequency changes over time, you know, so these ones are increasing, the blue ones are decreasing, right? So you're interested in these red ones because they are beneficial things. And so they fitted the density of distribution of fitness effects, basically. And so this would be the exponential distribution, which worked fine up to a certain point, and then they got higher selection coefficients, higher frequency of high selection coefficients, which they could explain in this case because that's your question. They had interference. Okay, what's your question? Substitution, you mean that got fixed?
Yeah, because I guess they have some way of accounting the fact that their power depends on x. So the very small less usually have much lower power to detect.
Perhaps, but I think the bigger problem, I mean, the bigger issue here, it wasn't the problem because they accounted for it, is that you have this clonal interference where different, how to say, well, you have mutations on different backgrounds that compete, and so the highest selection coefficient get favored. So you sort of have a push towards higher selection coefficient already through this effect, which is not a statistical effect, it's a POLYGENIC effect. Here, the large values, the higher values here is because, so it's not single mutants. You don't measure the fitness of single mutants. You measure a whole population where some of them had compete against each other, and this pushes towards higher selection coefficient, I think. Or basically, some of the low selection coefficient might have been eliminated.
When you have distributions with a logical upper truncation value, then the generalized Pareto gets weird and has shapes.
Yeah, I'm talking about that later. Yeah. Okay, so, but an important question, this is where I'm going to move slowly towards why you need the phenotypes, is that how does the DFE change with the environment, with background genotype, etc.? This is the big question that sort of eluded in some of this work. The background mid-fitness changes during adaptation. Usually, fitness is improving. So the new mutations, they arise in a different background with higher fitness later. So how does this change? This might modify the distribution of fitness effect. You might think that when there's more stress, it's more likely that some mutants might be beneficial. So anyways, this same paper that I showed you before, they looked at that. They looked at different environments where the wild type has different fitnesses. In different ranks also. They're not the best in some environments. The wild type was bad in the environments where the things have been selected because it was antibiotics and it was not resistant. But in other environments, it had different ranks. And so in this case, they showed that in these different medium, this kind of standard medium, then glucose with different, I didn't remember, I guess this is limitation of this sugar, or it's just providing this sugar, I don't really remember, sorry. Try different, sorry? The carbon source, yeah. So they try different carbon sources as opposed to a mixture in LB, perhaps? Yeah, okay. So it's kind of reducing to a specialized. Well, their message was that the distribution of fitness effects is somewhat unchanged and also with a similar mean. I still think that there wasn't very much data to estimate that, but anyway. If you want to have a bigger picture of this, you can do what they've done in this recent paper. So if there's anyone in the room who hasn't heard about Lenski's long-term evolution experiment, I don't know if I have to explain that, but anyway, they had 12 populations in limited medium, and you can correct me, with only glucose as a carbon source. And so that's it. And then it's been transferred daily since 1988, I think. So they have a number of generations that no one else can compete with. Perhaps if you start with the viruses now, you could catch up in sometimes. But viruses are not living things, are they? I don't know. Just to throw a debate. So they had these trajectories of increasing fitness over time. That's been studied a lot in a number of papers. And so what they did in this paper is that at different points, so you know, at 2,000 generations, 15,000, and 50,000, they did a random insertion of transposons, basically, yeah, to generate random mutations. And then they produce distribution of fitness effects through time. And so, well, the result was a bit surprising in that the distribution of fitness effects didn't change much. This is the ancestor distribution, the histogram, and then the blue line is the evolved after 50,000 generations. There isn't, this is in the log scale, so this means they had more than 6 lineage beans, contrary to the previous graph. But they did find some differences in the tails. And if you zoom on the tail, you see that the ancestral had an excess of deleterious, of highly beneficial mutations, as compared to this exponential, I guess, is the dashed line again. Whereas the evolved lost that. So there were more beneficial mutations, higher selection coefficients. And one pattern that you always see in these experiments, actually, so I've used these different experiments, you often see that there's this so-called diminishing returns epistasis where you have fewer beneficial mutations or weaker effect when your baseline fitness is higher. Basically, you have, when the relative background fitness is low, you have higher selective advantage, and then this decreases as you increase fitness. This is another experiment. This is yet another experiment, actually, but they looked at the final fitness and not the change in fitness. So if you look at the change in fitness, then it starts to look like the others. So this is kind of, it's not a law, but it's kind of very common pattern. So the same people that we saw earlier, they did a meta-analysis across several of these experiments. Yeah, there's E. coli, there's yeast, they find, so they call it the rule of declining adaptability. So diminishing returns is all kind of the same idea in microbial evolution experiments. So far, I think it's restricted to these microbes where we have more data on. But okay, so how do you account for these effects when you want to predict adaptation? You just focus on fitness and this distribution of fitness effects. So this is what I want to talk about now, where you use these phenotypic models where you track, the idea is to track the genotype and environment dependence of selection. And so one of the ways to do that that's become popular is this Fisher's geometrical model from in the same chapter as the fundamental theorem, actually. It's a few lines in the chapter. So it's an idea where there's an optimum for multiple traits, and this is characterizing adaptation in this way. And so Fisher's original argument was kind of an engineering argument about the curse of dimensionality, something like that. He wasn't very explicit about the fitness function or anything. He just said that if something about distance to an optimum is involved and you have a number of traits, then you have some properties that you expect. A mutation of small effect is more likely to point in the right direction and to not have overshoots of this kind. Yes? Yeah.
So you're saying that if there's more variance, I expect more change in the figures.
More variance? Oh, yeah.
If there's more variance, there's more change.
Mm-hmm. So you would like to interpret this in terms of changing the variance in fitness with the baseline fitness? You could interpret that in terms of the variance in fitness if you assume that there's an optimum like that, and then at the optimum there's less variance in fitness than away from the optimum. I think I'll go through that. I'm sort of eluding your question because it's not that easy to answer, but it might be later, actually. So yeah, what Fisher's argument was that the probability of improvement is higher when you have the lowest effect, the smallest effect, because they're more likely to go in the right. They're less likely to have these overshoots and other effects of this kind, especially when you have more traits. So as I said, he wasn't very explicit about that, but what he was explicit about is that he sort of established this equivalence between environment and background phenotype as a cause of maladaptation. Maladaptation might be because the environment has changed, or because the genotype could have a load of deleterious mutation or anything. And this equivalence is kind of permeates throughout this kind of theory of adaptation. And then it didn't have an explicit fitness function, as I said. So yeah, you can say that something is adapted only insofar as we can imagine an assemblage of slightly different situations or environments to which the animal would be on the whole less well adapted. Or equally insofar as we can imagine an assemblage of slightly different organic forms, which would be less. So I take this as placing this equivalence between genotype and environment. Perhaps that's not what he meant. So this has been later extended to include for the effect of drift, such that mutations have too low effect, cannot sort of overcome this drift barrier that we heard about, and so this pushes towards higher selection coefficients. But more interestingly, I think for us, it's been formalized and made a bit more quantitative to analyze how the distribution of fitness effects of mutations change across background genetic backgrounds and environments. And so this is the model that was developed by some colleagues actually in Montpellier some years ago, and it's a generalized multivariate model that's really similar to models we'll hear about in quantitative genetics, probably in the afternoon, where you have multiple traits. And so there could be the shape of the fitness landscape could be, so trait 1, trait 2, and fitness. There could be combinations of traits that have high fitness. And so there could be this fitness ridge that's aligned not along some of the traits, because there's so-called correlational selection that favors combination of high values at trait 1 and 2, for instance. So this S matrix is summarizing that. Then there's this optimum theta. And then from that, one thing that will be important is the fitness of the wild type, this S0 here, which is the maladaptation of the wild type, which you can get from the shape of the fitness peak. And lots of these models assume a Gaussian fitness peak. You could have more generalized forms. They would lead to slightly different predictions. And so a mutation with effect alpha has something that selection coefficient depends on its effect. This is like its squared effect, but in multivariate form. And then it also depends on how the optimum deviates, the trait deviates from the optimum in the background. And from that, if you have Gaussian distribution of mutation effects on traits, then you can say something about the distribution of fitness effects, because then you have a squared of something that's Gaussian, so it's a chi-square. That's the idea. But it's a generalization of that. It's a reverse chi-square, of course, because it's minus the square. And then it's a non-central chi-square because you have this deviation from the optimum, etc. There's more complications, but this is what it predicts. So it predicts that, let's see. Well, first thing I can say is that it's left skewed, just like in the data, and it's bounded by the fitness of the optimum. You cannot have a higher selection coefficient than the one that takes you back to the optimum that has selection coefficient S0, okay? And this is what I say here. There's this maladaptation term here, which depends on the fitness of the wild type. The model predicts that it will affect the variance of selection coefficients. This will increase when there's higher maladaptation. It will increase the proportion of beneficial when you are at the optimum, S0 is 0, you cannot have beneficial mutation, everything is deleterious. When you're away from the optimum, you start seeing these beneficials that can take you back to the optimum. Then the variance increases. The model also predicts that the mean selection coefficient won't be affected by maladaptation. This is in the Gaussian case. Then it leads to predictions that are more specific to adaptation, so beneficial mutations. That's what we're more interested in here. So you can get the distribution of beneficial effects, distribution of fixed effects. If you assume that the selection coefficient is 2s, this is a good approximation usually for small effects. Yeah. Is this intended for us, or I don't know? I was just saying that in this model, if you have a Gaussian distribution of mutation effects on the traits, which I depicted here actually. So, and I place them on the fitness landscape. The blue ones are the ones that get you towards these are the beneficials. When the wild type is at the optimum, there's none of that, okay? And then you have the deleterious here. When you increase this maladaptation, you see more and more of these beneficials. But then the average selection coefficient, including the deleterious, remains the same. But their variance increases. You see, you go from something like that to something that's a bit less skewed and broader. If you were very far from the optimum, you would get a Gaussian distribution. Very far from this optimum, this effect becomes this effect dominates, right? And then you don't have a square anymore. You just have something that normal distribution of selection of effects on traits sort of shows up. So it tends to a Gaussian distribution. Okay? So that's some predictions of these models. Lots of them have been tested actually at one point. But anyway, when you think about adaptation, what's interesting is that unless you're very far from the optimum, as I was saying, and you can sort of neglect some of these effects, when you're some way close to the optimum, then you're in another extreme value domain that's usually assumed. The reason is because you cannot have some beneficials that go to infinity as assumed by the exponential. You're stuck to not be higher than the optimum. So this predicts that there's, instead of this exponential distribution, there's this beta distribution. The distribution of fixed effect also has a beta distribution in this case. So this is a comparison in simulations to this exponential distribution to this beta in this case. So anyway, there's this set of predictions for distribution of fitness effects that can be investigated. Yeah. It depends on how you measure them. If you isolate individual mutations and place them in the same background, they're independent. But as it happens in the populations, it's not like that, of course, when adaptation is proceeding, right? If you're in the low mutation regime, then it is the case that you just focus on selection coefficient, fixation probability, then you move on to the next, and then that's it, right? And then this old theory of origination fixation applies. Outside of this regime is what we'll be talking about, where different loci, depending on their linkage, etc., they will affect each other, right? Is that what you mean somehow? Yeah, perhaps that's why we saw a deviation in this earlier slide where you have this excess of high mutations because they're not independent, I think. I think that's the reason. So anyway, you can also, I mean, all this theory just assumes that loci are all the same, or it's not explicit about loci, it's just genomic mutation effects. You can also have locus-specific distribution of mutation effects, and then in this case, what happens, what can be, usually it makes sense to think that loci, each locus doesn't necessarily affect all traits, or at least it may affect them in a very correlated way. And so you have this kind of restricted pleiotropy or modularity where the pleiotropy of a locus is lower than the number of actual traits under selection, right? And this produces these sorts of effects. So here it's a bit of a caricature, but in higher dimensions it's probably happening. This locus is stuck in this sub-dimension with just one axis, and this other locus is here. So it cannot access the actual optimum. It can access this own optimum where its subspace cuts the true fitness landscape. So each locus, for a given maladaptation, each locus had its own maximum selection coefficient that it can do. And so there's this causes some heterogeneity in their distribution of fitness effects that will depend on the level of pleiotropy, etc. So, and this, when you have such a context, this could be something that affects the probability of parallel evolution, how likely it is that in different replicates you hit the same locus just because they have different loci have different distributions of fitness effects. So, well, it's something I worked on a long time ago, but if people are interested, I can talk about this because then in a higher mutation regime, it's becoming less clear what happens when you have this. Yeah. I mean, it doesn't have to be. It's just that if you're saying at the whole level, at the level of the whole genome, that there are n traits under selection, but only m traits can independently mutate, right? Then effectively the dimensionality is just m, it's not n. If you have 10 traits, ideally that are under selection, but at the genomic level, there are only 5 that can mutate. Then the true dimensionality is just 5. You cannot evolve these 10 dimensions. But if you have 10 traits, but each locus can affect only 5 or 2, but overall they still have these 10 traits under selection. It's still the dimensionality of 10, but then you have subsets. So you understand what I mean? So this is why it's becoming more relevant in this case. If you have restricted pleiotropy at the genomic level, then it just means this is the actual dimensionality you're interested in. When you have restricted pleiotropy at a single locus, then the whole system, you know, when you're thinking of how it will evolve, how it will drift, everything that affects adaptation will really depend on the overall dimensionality, not the locus-specific one. You see? So, okay. So at this point, we say, okay, Fisher's geometrical model, okay, has its limits, etc. But it's one way to capture the context dependence of mutation distribution, the distribution of fitness effects of mutations. You have more beneficiations or higher stress. You have these diminishing returns. This is all the same thing, basically. Declining adaptability, depending on how you measure it. And so, okay, you could say, then this can be used to derive the trajectory of mean fitness over time by tracking the change in the distribution of fitness effects. Even when you don't care about traits, you have to have this layer of traits to predict the distribution of fitness effects. They could be very abstract traits that you don't measure. But still, if you want to predict the distribution of fitness effects later ones, you need this maladaptation, and you need to track the dynamics of traits. Also, if you going this way, this works best, as I said, in this sequential regime where you just focus on the selection coefficient of one mutation and you don't need to care about the rest. One mutation is sweeping alone, and you just care. That's why people working in this field, they just focus on selection coefficients, on fixation probabilities, basically. It's the main thing that matters. Escaping drift early on. But once you escape the drift early on, lots of other things happen, actually, when in a polygenic regime. What else? So, yeah, I mean, here, if you want to use this framework to predict, you need to know how the traits will change, to know how fitness will change. But I mean, of course, there's also arguments for saying that from that you can predict how the distribution of fitness effects depends just on baseline fitness. You can try to work on that. It's true. And there's also, there's some framework that actually have done that without Fisher's model, just try to find some, they call this global epistasis, and just the effect that it seems that fitness effects just depend on fitness, and you don't need to know more than that. But I think this model is more constrained, but it gives more direct predictions. I'll show some next week, I think, about how the environment is going to change all that, because so I think it's more complete picture, but I don't know if this answers the question. I think it's making it more difficult to predict things in other contexts, but perhaps not. I don't know. I'm just throwing this here, and we'll keep discussing that over the.
So here's the thought. So what you basically have there is a generalized distance measure from the optimal peak.
Yeah.
Doesn't matter what traits you measure, basically. There's some maximal aim you get.
Yeah.
That peak is always going to be shifting and flopping around.
Yeah.
So what about taking the largest eigenvalue of the large stochastic matrix? Does that kind of capture the same thing you're trying to get at? And that gives you a general trait independent measure.
So which matrix you're thinking of?
So your S matrix. Right, so your strength of selection.
Yeah. You're thinking this is going to fluctuate also?
Well, I'm just saying if you take a random matrix, like take a Wisher distribution and take the S as a draw from that, and look at its large eigenvalue, that might give you some idea of what that maximal S0 could be independent traits.
Yes, that's an idea.
Random thought.
Yeah. This is what, I mean, this theory was really based on, they use this random matrix theory a lot actually when doing that, but well, I didn't even come into changing environment. This is like in my next lecture, I'm going to talk about this movement in the optimum, but of course, this is one way of looking at things. There might not be an optimum even. It just captures well lots of the empirical features about distribution of fitness effects. Okay, so, but the point I reach here is that at this point, if you're willing to consider these types of things, it kind of makes sense to focus on phenotypes rather than just fitness, because it can improve the prediction for that. But of course, apart from that, it also brings lots of mechanistic insights if you're interested in what are the traits under selection. Obviously, if adaptation involves selection on traits, that causes an improvement in fitness. Okay, so lots of people have studied these modes of adaptation with an optimum, and when this directional selection usually is this shifted optimum, just one sudden shift. So it's kind of a minimal model of adaptation of directional selection on the trait. Biologically, you could say it's relevant for invasion of a new habitat. You can also have these critical transitions in climatic systems. Even there's something that looks like a trend, and then it passes some critical threshold, and then suddenly you have an abrupt shift, sudden exposure to antibiotic treatments. It's also just a very common, convenient way to just study a kind of response to a pulse or something, or an abrupt change. So again, you have these different regimes. So in the low mutation regime, you can have this series of fixation, and it's been described as an adaptive walk in this now famous paper by R. So you have the sequential fixations of mutations that approach the optimum, and they're fixing in non-polymorphic population. So in this regime, there are lots of things you can say, and actually R used that to sort of question Fisher's micromutationism, where he was saying you need mutations of very low effect in Fisher's model because they're less likely to be deleterious. Then Kimura pointed out that they need to be sufficiently beneficial to avoid being lost by drift. But then R pointed out that when you have this series of substitutions, the first one might be large when you're away from the optimum. And so the spacing, this was about the spacing of the first mutation and the second, etc. And so the spacing can be high in the first. So basically, the first mutation can have a very large effect on the trait because you're away from the optimum, and then they decline exponentially. So the phenotypic effects over the entire distribution trajectory, they're exponentially distributed also. So there are all these predictions that were made from this model, also still in a kind of abstract way that didn't really relate to DFEs or etc., but was more at the phenotypic level. And from that, you could get what I showed earlier, like this rate of change in fitness, this rate of adaptation based on mutational input and mean fixation probabilities, etc., but then translate it at the phenotypic level somehow. So one of the points was, in this paper, he sort of extended Fisher's argument about the dimensionality and the cost of adaptation by not just focusing on what Fisher did was the probability to be beneficial, but the actual rate of adaptation. And he showed that, the cost of complexity was higher than Fisher meant. Fisher thought it was in square root of n, but then here he showed that it was larger than n to the power minus 1. Larger than 1 over n, rather than 1 over square root of n. When you treat the whole trajectory, not the probability of beneficial mutations. So when you're interested in phenotypic change, there's also, of course, what happens usually is that populations, they don't reach the optimum. They enter this kind of drift zone or drift barrier zone. I don't know how to describe this. But basically, when you're at the optimum, if population size, effective population size is not very large at one point, you can fix some deleterious mutations that take you away from the optimum. Then you can have some compensatory mutation in blue that sort of, it's adaptation to the load of having deleterious mutations. So, and in the long run, the population will sort of drift around the optimum with a distribution that depends on the effective population size and also the complexity of the organism. So this has been studied in this kind of drift load, fixed drift load has been studied in the context of Fisher's geometrical model by using this phenotype distribution of fixed states. Okay. Now, when you move to more polygenic regimes, this is where we're entering the realm of this program, actually, and this is where I'll stop. Seems like I'm spoke too fast, I don't know. Now we're interested in the joint influences of mutation, selection, and drift in the establishment of alleles that contribute to the selection response. So this is like the gray zone that everyone's interested in. So how you switch from the sequential fixation regime to one where you have multiple things sweeping at the same time. And to do that, you need to focus on the establishment probability of rare mutations. And so there's been several recent papers on this topic. One of them by someone in the room, if I understood well. So it's not what I'm showing here. So this one is a paper by Joachim Hermisson's group. They assumed loci of equal effects. And so they focus on the probability that some mutations will occur, then they will escape drift and they will manage to sweep before the optimum is reached through the other loci. So you have all at a certain distance from the optimum. Let's say you would need 5 mutations to reach the optimum. So you have basically 5 loci that are redundant among each other. This is the level of redundancy that you have in this case. If you're farther from the optimum, then more loci could contribute. So there's more redundancy. That's how they define this redundancy. And this is the potential for polygenicity, if you want, during directional selection. If many loci, if different loci can be interchangeable, then this is opening the door for polygenicity. But it's not enough. You need enough mutations for them to co-occur and to start sweeping before the other ones have done it. So accounting for that, I thought it was quite elegant. They found that this single parameter, which is called the background mutation rate, so it depends on the mutation rate, on the effective population size, and on this genetic redundancy that I just told you about. Count the number of loci that can contribute. this single parameter, it determines how you move from monogenic to polygenic, going through oligogenic. So this is basically at a given distance from the optimum, the distribution of frequencies that you expect for these different loci. So for instance, here, you expect that one locus is fixed, near fixed, and the others are mostly at low frequency. And as you move on, you move on towards these regimes where many loci are at identical frequencies and they're sweeping together. So you move from this kind of sweeps to this kind of shift regime where you have small frequency shifts in multiple loci simultaneously. So this says that the response is likely to be polygenic is if there's large mutation rate, effective size, and large genetic redundancy. Of course, one of the big assumptions was same effect at all loci. This increases redundancy also. So when you further increase the level of polymorphism, you enter the kind of polygenic regime. Then you have many loci of small effects that can contribute to the trait. And then the genetic values will tend to something like a normal distribution, as in the infinitesimal model, which assumes like vanishingly small effects at many loci and no frequency change. So it's kind of an extreme limit. But actually, it's been shown by several people in the room that even in cases where you do have some deviation from normality, you get a decent prediction by assuming a normal distribution, or even one when the variance does not change because it reached some mutation selection equilibrium. There's an equilibrium between stabilizing selection that depletes variance, mutation that brings it back. So I think we'll have lectures on that. There are some regimes where you reach an equilibrium for this variance that's only caused by the stabilizing selection part. And so you can sort of neglect changes in variance, and you can apply well, this Breeder's equation, but in evolutionary biology, we rather use this type of formulation where you have this selection gradient, which is the slope of the fitness landscape, times the additive genetic variance of the trait. And so this is nice because it kind of uses the formalisms of Wright's fitness landscapes on allelic frequencies, but extended to mean phenotypes. So in this recent paper, they showed that, so I believe you're the first author, right? Okay, so you can correct me if I'm wrong, and there's lots of things to this paper, but one of them that I found interesting was that for most of the course of the approach to the optimum, you get a decent prediction by just using this assumption that there's constant genetic variance. And then what's nice is that you can sort of have very simple analytical results like exponential decline of the distance to the optimum. That's what this predicts. Since the selection gradient for Gaussian fitness peaks is just proportional to the distance to the optimum. So the change in the distance to the optimum is proportional to itself, exponential decline. That's simple to understand even for someone like me, for instance. So it works well up to reaching close to the optimum where you start having more effects of stabilizing selection that matter and drift, etc. So in this regime, whether it's polygenic, all frequency changes are small in the short run, and they're proportional to their contribution to the genetic variance. And actually, even if a locus has a major effect on the trait, so this one could have a strong selection coefficient, it's still standing on the genetic background, right, with mean phenotype Z bar. And if this mean phenotype in the background is evolving because of all these other traits, its selection coefficient, which is proportional to the selection gradient, will also decline over time because the mean phenotype is approaching the optimum. And so this will cause a decline in its selection coefficient because of other loci that are evolving in the population. In some cases, this could even lead the beneficial mutation to become deleterious if you have this mutation with effect A, or well, 2A in homozygous state. So it pushes the mean trait in this case. But as the mean background is evolving, approaching the optimum, now it's become deleterious over time. And so because it overshoots the optimum. And so in this scenario, you could have just start changing the initial conditions a little bit, or changing the effect of the mutation on the trait. You could either have this loss of mutation where the mean background phenotype reaches the optimum, or fixation of this allele that affects the trait, and then the mean background sort of evolves to compensate for that, and you have such that their sum is at the optimum. So this has been studied also quite a lot, this type of scenario, by Kavita here. She probably talk about this with more explicit loci, etc. Anyway, I think I'm going to end up with these considerations that in this highly polygenic regime, each individual locus has little predictive power. Plus, it might have complex dynamics, which we'll see in next talks, I think, where it's becoming a bit difficult to predict. So if you don't want to focus on single loci, you could have this genomic selection type of approach where you predict based on the whole genome genotype. The way I understand it, you need to have reliable GWAS, genome-wide association studies, to do that. So the problem with GWAS is that they're very population-specific. No? You're saying no? Okay. Yeah, genome-based heritability. Okay. Yeah. Anyway.
We'll talk about GWAS.
Yeah, we'll hear about that. So I'm just throwing away keywords and for other people to be more precise about. But it seems like it's very population-specific. You find the genetic base of a trait in one population, then it's difficult to predict what will happen in the next population. So people have tried to find so-called saturated GWAS in Peter Visser group, and I think they had sample size that you can only get in humans, 100,000s, in the 100,000s. Oh, 4.6 million. And so they said they had the set. So if you need if you start needing the same order of magnitudes as the whole species to predict, is it still a prediction? Anyway. So, and then old school quantitative genetics just tracks the moments of phenotype distribution without attention to details. And if you're willing to take some of the wild assumptions that are made, what's interesting is that, such as these ones, you don't have to assume that, but if you assume that, then you can use that to make analytical progress in some explicit scenarios of adaptation where you start changing environments, etc. So that's what I'm going to talk about in my next talk. Of course, then it's also not satisfying in some respects because it's become difficult to confront to some types of data. But yeah, I think it helps sort of focusing on scenarios of adaptation that are a bit more complex at the cost of not knowing much about the genetic details. So that's it. I finished a bit early, so I talked too fast probably. So if you have questions, don't hesitate.
This opens up kind of a general theme I think you'll get through here. And that is, are your models better off when they're highly specified or very fuzzy?
Ask the audience.
Basically very fuzzy, right? We have a variance. We kind of don't care how it gets here. And we know it works because we've got, 100 years of agricultural progress that does do something. So one of the things we'll talk about is what we gain from all this additional genomic detail.
Depends on the questions, of course, right? But it's a question in itself to know how genotypes change, right? But if that's not your specific question.
Just another quick comment. So one thing that hasn't come up is the red cleavage. So Van Balen had this notion that because populations are always evolving, if you stop evolving, you get left behind. And that's kind of like what that means is your S0 is probably never goes to zero, because as it does, you keep on getting further and further behind, which means you're always at that beta distribution realm where you've got that truncated positive S values.
I think the question you raise more broadly is about how can we trust equilibrium situations? And we all rely on that. Even in modes of moving optimum, you have to do a burn-in, or you assume some long-term equilibrium with a stable optimum before something changes, because otherwise you cannot predict anything. But it's very difficult if something is always, or you have this kind of stationary fluctuations, and then you can say something. But in all situations where the patterns of change are going to be very variable, I don't know what we can do, but we always rely on some equilibrium assumptions, right? Usually. I don't really know how we, myself, I don't really trust things, I don't really believe models or theory that doesn't have any environmental change. But perhaps it's relevant for some, yeah, it's very relevant for like internal traits, like cellular traits or these types of things. For other types of traits, I don't really, like this, for instance, this very long-term mutation selection drift equilibrium. I tend to think that any change in the environment would perturb that. See what I mean? So it's kind of related to what you're saying.
Yeah, because I mean, your conspecifics are your environment, and as they evolve your environment.
Oh, yeah, then there's that, but this brings the frequency dependent things.
When you talk about a trait that's something with general fitness, right? You can imagine a trait running in some physiological limit, or the environment not impacting it so much as you get this alpha value. But if your trait is competition, there are lots of things that live there that's always shifting. So Nick may have better metabolism than I do. You know, all of a sudden I get a little bit larger, and he's going to get his metabolism up, right? So you see that this.
Yeah, for fitness, it's obvious that it's not ever increasing, right? It's obvious that.
Well, it's not ever, the relative never increasing per se. But if you, I mean, the idea with red cream is I can take an organism and put it back into the identical environment 1,000 generations ago. It would be a superstar. Maybe that's true. That's the logical assumption of red cream.
Well, this transitivity assumption, but it's been tested by some people. It doesn't really. It's either the kinds of arm race or like I think red queen is kind of the reverse. You're better on your current one, but not necessarily the past or the future one. So people have tested competition with present, past, or future in some either experimental evolution or a resurrection ecology where you get seeds from the past or things like that. And I think yeah, it's both. But actually, something like this is the case in Lenski's experiment. He's advocated a lot for this use of relative fitness to the point of comparing something to its ancestor 50,000 generations ago. It never competed against that. So I'm not sure it's more meaningful than just the absolute fitness. I don't know. In this case, they tested for transitivity nearby, but not like, what does it mean? Yeah.
The
50,000 generations. I mean,
I don't know when they have done it last time.
key is a comparable environment. That's the thing. So the key is testing in a comparable environment. So, for example, maize production in the U.S. has gone up dramatically. I think Mike showed a slide that early on. Almost all that gain is not because individual plants are getting better. It's because you can pack individual plants close together and have them keep their yield. So they're getting less.
But this is a response to selection?
Right.
It has been selected for that, or?
Yeah, there's like these basic leaf angles you can pack them closer so that each individual plant has the same amount of seed. Now you put 10,000 plants and make the instead of 5,000.
So it's indirect genetic effects or these types of things, is it? Indirect genetic effects, these types of things?
Yeah.
It's a good question to what extent you have an view of this, or to what extent you have basically trade-offs of specific adaptation. I don't know how much you know.
No, but even in the genetics literature, people are trying to do that, to know, well, trying the fitness effects of mutations in different backgrounds. Yeah, there are people who are studying how the correlation of the fitness effect across backgrounds, for instance, these types of things, you know, and so that's one way to look at that. If you look at different genotypes in the past over adaptation, does the selection coefficient change a lot? Is there a directionality to that? Is it autocorrelated?
Well, they're doing that correctly with resistant genes in agricultural species with CRISPR. What you find out is in some background they can work not unexpected.
But it's more the direction, I think, of the mutations, like the order of the mutations.
Well, a Fisher model would predict that the epistasis between two mutations doesn't depend on which background they're assessed in. So this is a big prediction that's not necessarily, I don't know if it's verified, honestly. At least you have a prediction. It's epistasis between two mutations, but in different backgrounds.
I know, but it assumes that these two mutations have no aesthetic interaction with the other background.
Yeah, it assumes, because it uses the level of traits, and so it assumes that their phenotypic effect on traits don't depend on the background, for instance. So there's no epistasis at the phenotype level. The assumption could change that.
To what extent do you think it's favorable to study the phenotype of fitness? What do you favor?
I think in some places, lots of people cannot measure the phenotypes. Like lots of people doing microbes, they measure fitness because that's what they can measure. It's difficult to measure individual traits. You can, but I mean, so.
So is it better to work with, I don't know, Latin, where you can measure phenotypes? Or is it worse because you don't know what phenotypes?
I don't really know. I don't want to. I mean, I think different systems have their specificities. It's just actually my impression was that the whole conference would be about phenotypes. So I actually wanted to talk about people who don't measure phenotypes. But I know that, I mean, it's a very, very difficult endeavor. I mean, measuring phenotypes is so much more costly than just sequencing, that no one wants to do that. Knowing that you measure the relevant traits, how many you need to actually cover adaptation is very difficult to know. I mean, it's
Just to say that in the world population, measuring fitness is much harder than measuring traits. Fitness, you need to know, the offspring, phenotypic environment,
yeah. You need to make sure people agree on the way you measure fitness, that it's actually fitness.
But the flip side is
another trait in measuring fitness proxy.
But the flip side is to measure fitness without traits, you can bound the maximal change in any single trait without further selection. So that's an advantage to measuring fitness. You can measure it. You just need many generations.
So my point was just that if you focus just on fitness and you're dealing with adaptation, there's this big topic of how the distribution of fitness effects change with the background fitness. So you can study this phenomenologically, and you see that it happens. You have these curves, it happens all the time. And so it's more satisfying to me, it's more satisfying when you can explain that, because sometimes verbally, people always talk about optimum. But, oh, what's the optimum phenotype? They're always mentioning that. So it's kind of good to make this explicit and see what this predicts. What these people have been doing, and I've worked a bit on that, but it doesn't mean you need this necessarily. And also it gives a model that you can test and try to reject. In some cases, it's not that, but this one, it's been tested. I mean, I'll show some other things next week. Lots of aspects have been actually tested, like that the variance of fitness effect changes with the background, these things about epistasis. There are lots of things that have been tested. So, but perhaps it's right for wrong reasons. I don't know. So, yes. Yeah, this is what Nick was saying also. Well, I mean, when you have a multivariate setting, it's sort of arises by itself. You know, if you have this, at least it would produce that. If you have the genetic correlation and correlation and selection, you could see evolution going in one direction, then completely switching just because of the shape of the fitness landscape. So this you would predict, but the thing where what you have in mind is perhaps something more ecological, where you have some kind of, after you change this trait, then you produce the molecule or something changes the selective pressure completely. The traits are different. Yes, all this happens. It's the ecology, I would say. It's difficult to treat everything, but to me, I think it's interesting to keep all these things in mind, even if you're not studying them, right? If you have an organism where you mostly measure growth rates and things and try to think this is underlain by these types of things, might help you think about this better, I think. And like, yeah, I don't know, there's theory that just assumes there's a steady input of beneficial mutations with a constant distribution. To me, it doesn't, I don't really trust this much. And this theory has been less confronted with experiments, actually, than these things I'm mentioning. Okay, I think we're ready for lunch, no? Yes. Thank you.